Right, morning everyone. I think we're still waiting for a few people to arrive and we're a little bit early, so I'll let you all have your morning coffee and we'll get kick-started in a minute. I have a small apology to make about our pestivirus lecture. It's somewhere in the Zoom cloud and we're all trying to get it off the Zoom, but it seems to be evading even the most techie of people. So I think I'm probably going to re-record it for you guys because we've exhausted all of our usual routes of trying to get it off. Oops, chatting. In terms of the local hard drive, these Zoom sessions are set up so it should record anyway to the hard drive, but it doesn't seem to for some bizarre reason. And so we're figuring it out. At the moment, we're all learning about the weird foibles of Canvas in Zoom. So we're trying. Even Jenny, who's like a super tech cow vet, can't figure it out, but we'll get there. How are we doing? Everyone's still waking up. We're getting there. Look. Have a look at everyone's pictures. We've got some cats. Lucy has managed to wake up this morning. She's come to join us. Morning, Alex. You smile. It's a bit early for you. Yeah. Oh dear.
She's adorable. Oh, look at the paw. That's so sweet.
Thanks. She's unfortunately she's a little bit spoilt and she has to be patted all the time. So I'm going to pat and lecture at the same time. Hopefully that'll keep her happy. Good girl.
She's too adorable.
Oh, come on, closer. There we go.
Well, this made my day.
Oh, thanks. I like seeing everyone's picture of their cats. It's good. We've got a little few tabbies in there. Jenny still has one surviving kitten, everyone. We think her other one's got a very, very, very nasty dose of cat flu, which they were struggling with when they arrived with her. So
the other kittens die?
2 of them did, sadly. Yeah.
Oh, I didn't know that. Oh, that's so sad.
Yeah. So it was really sad. Morning, Hannah's dog. It's got a little jumper on. Look at that. Oh. So we've, yeah, she's got one left and it's bright as a button and it's tested negative for parvovirus, which is really good. So it's one of the really, really fluffy charcoal tabbies is still alive, which is...
Is that the kitten that Ripley hid? The one that was covered with drool?
No, I don't think it was. I think it was the other one. So whether she was like trying to save it and...
Yeah, Ripley apparently took one of the kittens and hid it in his special place.
Yeah, no, Ripley's little cave is quite impressive. There's lots of funny things in there. Right, how are we doing? There's a few more people waking up. That's good. Right, so this morning we've got Yoni's disease. Now, when I took over these lectures, they originally were 2 lectures of an hour long, and they've been squished down into an hour. So it's going to be a whistle-stop tour of all the important clinical things. Yoni's disease is a really, really big subject, and there's loads of information out there. So I've put you down some little resources on Canvas. I've not made them compulsory reading, but I have put down whether they are light reading, easy reading, whether they are more kind of technological, and whether they're applicable to different areas of the country. And again, if any of you fall asleep or have to go have a coffee halfway through, I put little stars on the pages that are really important. And they're the kind of things that we like to take exam questions on. Okie dokie, shall we get started? Go and share my little PowerPoint. There he is. Right, I've got the chat box open again. So I'll try my best to keep answering things as we go along. Right, Johne's disease. This is one that crosses a couple of our different categories. It's basically all the ruminants that we're going to see here. I'm just going to move my chat out of the way. There we go. So we've got our little learning outcomes, and these are my two goats who are dancing on top of the fire pile. We're going to think about how we're going to control Johne's disease in cattle. So BJD is bovine Johne's disease. And we're also going to think about how we're going to describe the advantages and the disadvantages of the diagnostic tests we have to help us use bovine Johne's disease. So just to give you a little bit of a signage, we've got our etiology, pathogenesis, some epidemiology, all about the clinical signs and our differentials that we might have to consider, and any treatments, controls, and our public health considerations. Now, I showed you cow of the day on our pestivirus lecture. This one here is actually my favourite cow. This is one of my dad's cows. She's called Dora, and she's a Belgian Blue Cross Longhorn. She's quite photogenic. I like her very much.
Victoria,
Yeah.
I'm not sure we can see you switching between slides.
It's running on mine. Let's have a look.
I think you've got the wrong screen shared. Maybe we can just see your original PowerPoint slide in PowerPoint.
In PowerPoint. Let's escape from that. Right, what can you see now?
Transmission host interactions.
Yeah. Yeah, that's right. So if we go back one. Can you see etiology now?
Yeah.
Yeah, that's okay. And is it flicking slowly or just not flicking at all?
It's still an etiology.
That's fine. Yeah.
Yep.
Yep, we're all good.
It wasn't flicking at all before, that's all.
All right, well, we'll shout up again if it decides to slow down.
You might want to try the presentation mode to get full screen.
I think it's in, well, it was before. Is it really, really slow? Like, it's in full screen now.
It still
Can't
works.
see the full screen version, that's all. Like, we couldn't see it when it was in presentation mode.
I'm good to. So the moment I've got. Is this what you guys can see? A whole screen.
We can see that's, we can't see the whole screen like that. We can just see it still in the slide
show
kind of setting, yeah.
I wonder if we've got a dodgy internet connection because it's currently running.
Can you just have it so you just flick through the slides on the slideshow instead of the full view, just so we can...
We might do that because... Right. I'll move that down a little bit so it's bigger. Shove you over as well. Right. Okay. Can we see?
That's good.
Yes, thank
You
you.
good? Right.
Yeah,
We'll go with that. Okay. So we've got our distinct cattle and sheep strains for this. So C and S is what we usually term them as. The full name is Mycobacterium avium subspecies paratuberculosis. Because it's so long, everyone always calls it MAP. And it has to live inside its host to replicate. In terms of how we see it when we're doing pathology, we can use acid-fast stains, and it gives that lovely red colour. And it likes to use mycobactin to help it acquire iron when it's inside the host. So it really needs that, otherwise it's not going to replicate. It's very, very slow growing. these take at least a year and a half before you will start to see any sort of humoral or cellular mediated immunity or any clinical signs. So whenever we see any clinical animals, they are the tip of the iceberg. There's lots of others who are still undergoing the very slow process of acquiring clinical infection. In terms of transmission, we have a small number of hosts that we find in Australia, and it keeps things fairly simple. So the ovine strain just goes round between sheep, and the bovine strain goes between cattle, and it can sometimes break out into goats and deer as well. So you bear in mind, with the type of populations we have in a lot of the country, we're definitely going to see a lot of deer, a lot of wild goats around. So in kind of around particularly Sydney and the western area, around the Blue Mountains, it's a big concern to us. There's loads of extra hosts around. In terms of globally, the picture gets much more complicated, and the strains hop all over. There's not often many sheep strains that going into cattle, but there's a lot that come the other way. So basically, we treat everything as a possible host and a possible risk factor. The pathogenesis is super, super slow. So this gives you a little bit of an idea of all the things that are going to happen from the first minutes that a neonate ingests feces that are containing MAP, all the way through to the months where they're an adolescent, and to the point of years old, when you actually start to see shedding of MAP in the gut and responses by the body. So kickstart by ingesting it, and it goes through the digestive tract and sticks to the epithelium. There, it's pretty clever, and it overcomes all your defenses and gets eaten up by the macrophages. Unfortunately, it doesn't get killed by them, and so they use it as an Uber. And so our macrophage Ubers, they're going to take it around further in the digestive tract. So it's got its mycobacterium to help it get some more iron, which is essential for its growth. And it then starts to hop out of its Uber and localizes in the lamina propria of the gut. And that's where we're going to see it if we're going to test for it in post-mortem samples. So this point, years down the track, we're going to start to see some cell-mediated immunity, making cytokines, and also some humoral, making antibodies. And finally, we start to shed it from our gut lining. So here we've got some cattle post-mortem samples. We've got some specific areas that are really important for where it localizes. So you can see that they get a thickened terminal ileum. We have enlarged mesenteric lymph nodes, with a bit of a dark middle to it. And we've got all these little stripes of white where we've got lymphangiectasis as well. So these are quite easy to pick up if you're used to seeing normal pathology.
Hey buddy, how you doing? Oh my God, I'm so sorry.
That's all right. Lucy's just popped in as well. And this is what it looks like on the inside. And so if you imagine a normal ileum should look a lot like a balloon animal. So really thin lining, nice and small and tubular. And this ileum becomes really, really thickened and greasy, and it's got lots of roughened areas as well. So almost it becomes like a form of, you know, when you buy a supermarket chicken and it comes in a plastic tray, and underneath there's a little pad that the chicken sits on, and it's all kind of slimy and thick. It's a lot like that. It's the feeling of like this chickeny sanitary towel almost. It's the kind of anecdote you'll never forget, that one. In terms of sheep, we'll see again our lymphangiectasis, and we get this thickened ileum as well. But as you can see from this slide really nicely, they've got normally formed feces. On histo, we've got enlarged Peyer's patches. We don't often get to this kind of stage because the early stage is been and gone by the time vets are called to see them. So this is kind of a rare thing we will actually get to look at. And pathologists will use a Zeal-Nielsen stain to find these red acid-fast rods. They're really pretty and inside all of those little villi. So in terms of histopathology, we've got a normal ileum on the left-hand side, and we've got lots of really nice long villi and big crypts between them, and they're great for absorbing water. However, if you've got Yoni disease and you're clinically affected, you'll get to the point where you have pretty much no villi left. They're all blunted, there's hardly any crypts, they're really, really small. And there's all this granulomatous inflammation as well. You can see loads of blue and red, lots of cells in there. And these kind of give us an idea of the clinical signs we're going to see. So we're going to have lots of profuse diarrhea, because we're not absorbing the water. We have no capacity to do that. And we also are going to be losing albumin through this as well, because we're not able to absorb anything. So in terms of pathogenesis, this is a really, really slow plod disease. It's going to affect your macrophages. You get clinical signs at the very end of the disease due to the immunopathology. We'll see diarrhea in cattle and not sheep, and we'll have a bit more of a chat about that. And our weight loss is from all that. Those little villi are very, very small, and we're losing lots of protein. Epidemiology, this really comes very much with the young animals to start with. They're the most susceptible. Cattle are partially resistant to the sheep strain, so sometimes it's safe to graze sheep and cattle together. However, our pathogens are really resistant in the environment. It's very difficult to clean an area that's been infected. There are hardly any disinfectants that are going to kill this. But, as in the environment, shaded areas favour the survival of it. So UV light, so everywhere in all these amazing sunny pastures in Australia, they're going to kill the MAP bacteria quite easily. But anywhere else, so here you got a shed. So in the Northern Hemisphere, we keep a lot of our animals inside over winter. This is a great area. There's not a lot of light coming in. It's going to be really, really hard to clean that of MAP. And in terms of your flock management, if you've got a lot of animals all together with their calves or lambs, then you've got a high exposure rate. If they were out in the pasture and there weren't very many of them together, then you're less likely to get exposed because you're in a greater area. So our sources come in twos. So our transmitters are all of our ruminants, and we can find it in lots of environmental sources, and these are always changing. It's a dynamic environment, particularly if you've got water contamination. So you think of creeks running through properties, rivers running through, different pastures and different soil types. And as the weather changes, amounts of MAP will die off with more UV light or come back if you've got lots of very shady days or lots of trees places. Our fecal oral route is our major route. That's the biggest concern to us. And it's from the adult animals shedding the MAP in their feces and the little neonates coming along, suckling on their mother's poo-covered teats or nibbling it from the floor by accident. Intrauterine infection is also one of our concerning routes, and that's quite hard to often pick up. And transmammary as well, so from our milk and from the surface of the udder. Usually, quite often it's spread in short distances. That's the most common form. So you've got one infected property and everyone's kind of just starting to spread outwards. Long distance by trade can in some instances be a big concern, particularly if we're not very good at tracing animals from one place to another. And this was a big concern back in 2001 in the UK. We unfortunately had a very large foot and mouth disease outbreak, and thousands of animals had to be euthanized. And And after that, there were large areas of the country with no cattle or sheep at all. And so they brought in animals for other areas of the country. And unfortunately, people didn't have a huge amount of choice of what they bought. And they bought from other areas into their own map. And so Yoni disease spread across hundreds of miles. So in terms of transmission, we vets like to call it poo in mouth disease. And here's a picture of our major source is from the environment where it gets pooed out onto the grass and the young ones eat it. And then our minor ones are in utero infection. In terms of cattle, we've got a little bit more complicated scenario. Still, our major one is poo on the pasture and on the mothers. High stocking rates will affect this, so specific calving paddocks, particularly in dairies, where they'll use the same calving paddock all year round. And if one cow with MAP is shedding, then all the calves born in that area at that time potentially could be exposed. For a lot of farmers, they'll only see the clinical disease when they've got diarrhea. And this is the tip of the iceberg. This disease is so slow that there are so many other animals who are in the younger age bracket will have this that we can't see and we can't test for either. 22% of our subclinical infected cows, so those who are under the tip of the iceberg, they will be shedding it in their milk, so it'll be going into their calves. And 40% of our clinical cases and 8% of subclinicals will have infected their fetus in utero. And this is quite a big consideration for some of our clients where they know the cow isn't well, but she's heavily pregnant, and they would love to at least keep the calf if they know that at some point they're going to have to euthanize the cow and before her welfare becomes a concern. Unfortunately, the ship has already sailed. That calf is probably going to be infected as well. Incubation is really, really long period. We've already talked about how slow this grows. And fecal shedding is the first thing that we start to have. And that can happen for several months before we see any actual visual signs that the animal is unwell. So we're always playing catch up with this disease. Funnily enough, MAP itself, I'm just looking at Jess's question, MAP doesn't affect the bulk cell count, but these guys, because their immune system is under attack, they're more likely to be sick animals. So they're more likely to have mastitis cases that are recurrent or a grumbling high cell count. They're more susceptible to getting other problems. And we'll talk about human consumption at the end. We've got a good slide. So in terms of when they're very little baby calves and they become infected, it can take at least 9 months. That takes them through all their kind of childhood into adolescence before they start shedding. Unfortunately, we can't test them until at least 18 months old. So they've still got some time where they can be shedding and we don't know about it and we can't test for it. And they look perfectly fine. It's only when they get older that we start to see the clinical signs. So it's really adulthood is our main ballpark. So sheep is over 1 year, cattle is over 1 year and basically for their whole lifespan. And really the time at which you start to see clinical signs depends on how much MAP they ingested when they were neonates and whether they were very much baby neonates or whether they were a little bit older. So in summary, we've got that a lot of it is direct contact with the young, with the old. And we talked about the tip of the iceberg. So 25% will be our clinicals. 75% is all of our subclinicals that we don't necessarily know about. It's very hard to find them. So it means our prevalence is difficult to estimate. Basically, if we find a clinical case, then we know that this is definitely going to be something that's spread between a lot of animals. And so in terms of what does it look like, this is a dairy cow with it. She's losing weight. You see a little pin bone there. And she's got profuse watery diarrhea. They don't have any tenesmus with this. It just literally falls out of them without them even trying. Farmers tend to first notice that they start to reduce their milk production. And even with extra feeding, treatments for worms, and investigating other causes of weight loss, they still find that it's still losing weight, still producing diarrhea. And these guys can usually succumb quite quickly. From when they produce the profuse water diarrhea, they will only last a few months. And when the farmers know they've got Yoni disease on their property, they're quite quick to identify them. Sophie's asking, do all subclinical cases eventually become clinical? Yes, at some point, but for a lot of our farm animals, they might die for other reasons before they become clinical. So they might be culled for lameness or poor reproductivity, mastitis. Or if they're beef animals, they might get old enough that they're able to be eaten. So that means we often will miss them and not be able to test them either. There are very few that will, if they were left to live their full lifespan, would go past that stage and never get identified. Sheep are a little bit different. They will not display diarrhea, but you'll see the weight loss, and as much as people feed them and drench them for worms, which are one of our kind of major concerns for weight loss in sheep and goats, they're not going to improve. So we see these skinny goats and skinny, unhappy looking sheep. So cows get diarrhea and sheep don't. Some of our differentials. We've got some differentials for emaciation. All of these are fairly sporadic. You're not going to get a lot of these coming through a large number of adult animals. And they don't normally give you diarrhea as well. In terms of diarrhea itself, we know that all of our clinical signs are going to be in older animals, and these ones tend to be more for young ones, or affect whole herds at once. And we're looking at individuals drip by drip, one after another succumbing to this disease. For small ruminants, we've really only got emaciation as our main differential to discuss. And some of these diseases, thankfully, are exotic to Australia or occur in small numbers, and will be quite sporadic. Some of them are quite easy for us to tease out as well. So we can do fecal meg counts, we can do clinical exams, we can know about the land itself and what kind of deficiencies there'll be. And malnutrition itself, you'll see over a whole number of animals. So if you guys have seen the pastures and the animals the last few years where we've had horrendous droughts, that affected all the animals on the properties, not just the old ones. So here's our diagnostic tests, and unfortunately, the majority of them are absolutely rubbish. It's very, very hard to get a one-shot successful test, mainly because we've got to wait until the body is responding to this disease before we can start looking for those markers of disease, and whether it be we're looking for cytokines, whether we're looking for the MAP shed in the feces, or whether we're looking for antibodies. And the aim is that we start testing and we keep on testing. We've got to regularly test animals. The more we test them, the more likely we are to catch it, because we can improve our specificity and sensitivity. So for feces, we can test the individual or we can test a mob. So we can go around at milking time and collect from the milking yard, and we'll get a good proportion of the cows from the herd. We can smear and then do our Zeal-Nielsen test, looking for acid-fast rods. Most often we will culture. It does take 13 weeks though to get an answer, as they slowly grow in the lab. So this takes a lot of patience, and you've got to tell your owners that they're going to have to wait for the answer. We also have a high throughput Yoni's PCR. This is really handy as a quick initial test. And for some clients, it means that they get a bit of a snapshot idea, and it gives them something to think about while they're waiting for their culture results. In terms of blood samples, this is probably one of our mainstays. So we're going to look for antibodies with an ELISA. We can also do additional tests as well. We might do this more with individual patients in backyard scenarios. So we can look for low total proteins, hypoalbuminemia and hypocalcemia. So we're not absorbing a lot of our nutrients and proteins. And we can also do lithet tubes for gamma interferon tests. We don't do this very often. We tend to stick to our plain tubes and the ELISA. Not a lot of places will offer the gamma interferon test. It's more used at a kind of government level. In terms of biopsies, obviously we've kind of missed the boat if our animal has died or we've euthanized it, but we could take samples of our gastrointestinal tract and submit those for histopathology. The abattoirs will also keep an eye out for this, and although it's not going to affect the carcass, it is something that we can use as surveillance tool. So I've got a few different diagrams, and it helps to give you an idea of when we can test, why they don't necessarily work so well in the younger animals, and gives you an idea of specificity and sensitivity as well. And depending on how you like data or looking at graphs, one of these will suit you. So here we've got from the point at which these neonates ingest the map, and we're going right over to the months and down the line to the years. Now at this point, we're going to start seeing the bodies responding. So we've got Steph's asked about hypocalcemia. We're not able to absorb our calcium. So those little blunted villi can't get in. And if we're losing protein as well, then it's going to go with them. And the protein is not able to be absorbed either. So both calcium and protein are going straight out the back end. In terms of our cell-mediated tests, when we get to kind of adolescence, it's probably the first time when they're at least 18 months old that we can start to test. We don't use this test very often. It's not provided by a lot of places. But you can see there's only a short window that we can test them. And it starts to go down towards being a negative response by the body. So our fecal culture and our antibody tests are our mainstay. As we get towards clinical disease down here, the likelihood of getting a correct positive on fecal culture increases, and the same with antibody tests as well. But really, you're getting towards the end of the line once your antibody tests become proper, true positives. There's no going back from that. So here we've got another one. So we're going to go down the cascade from a little neonate being infected. They'll sit with a silent infection for many months until they're becoming an adult. Again, our cell tests are going to give us some good positives at this kind of teenager stage, around 18 months. And then as they start to shed map in their feces, we can start to test for that with fecal cultures and fecal PCRs. That's our first sign. After that, we're going to get clinical disease, at which point we can start doing antibody tests, and we can also do our pathology tests from postmortems. So here's a little site for all of you guys who really, really like the very techie side of things and sensitivity and specificity. And it goes through all of our tests, and you can see at the bottom here, our sensitivity and specificity aren't that great. So the more that we do these tests, so whether it be we test cattle every year, or for older animals we test them every 6 months, the more we test them, the more we can improve these values. Oh, why is it stuck? So our cell-mediated test is going to look for cytokines, and that's with our gamma interferon test. But really the government's the only people who tend to use that. And you've got a very small window to get it at. So it's quite difficult for us to test. It's very hard for us to plan at which point we're going to try and use that test. They're usually quite expensive as well, which is why you don't see them used in clinical practice very much. I'll come on to the finances at the end because I've got a little slide on monies. In terms of limitations and benefits, advanced clinical disease with all that diarrhea and weight loss is very easy to detect. Before that, it's very, very hard. We can test, but we don't really know who to test unless we test everybody. We have very, very poor sensitivity in testing for that early disease. And so we tend to just say, in order for us to get accurate results as best we can, we don't test anybody who's less than 18 months old. In terms of benefits, for everybody, it's pretty easy for us to figure out what we're going to do in terms of testing. Whether you're testing individuals, whole herds, for accreditations, or for regional surveillance, it's all the same samples. So whether it be blood and feces are the ones that we're going to try and take. And if we have a denouement, it's post-mortem specimens of the ileum. Now, unfortunately, treatment is not really much of an option. By the time we know these animals are infected, it's almost the end of the road for them. And treatment is rarely indicated. It's occasionally been done in research scenarios, but the drugs that are required are not necessarily licensed in livestock, and also they're very expensive and unlikely to give you a significant outcome. They do use them in humans for Crohn's disease, and that's kind of where our shared thinking has come from. But unfortunately for these guys, we don't tend to treat them. And really got to think about the bigger picture. Control is our most important tool. So we've got 3 principles. We're going to prevent it coming into our herd. We're going to limit the transfer of it between our animals if we do have it. And we're going to remove those infected shedders, the ones who are actually spreading it around. So a bit similar to pestivirus, we're going to have fences and biosecurity. So we're not going to be able to throw that poo around, poo over a fence so another one can get to it. If you've got double fencing with a 2-meter space, it's very difficult for you to launch your feces over to the other side, to a naive herd. And we can run closed herd policies. So everyone in that group, we use just them. We don't buy in anything new things that might be infected. And if we are going to buy things, we can buy it from accredited free herds. Biosecurity is important. So if we're going to go from farm to farm, we make sure that we clean and disinfect. Now, I know disinfectant is not necessarily the be-all and end-all with this disease, but at least if we can remove the frank feces from our clothes, we're at least trying to make a difference. In terms of transfer, we can manage all of our situations. We can try and reduce the amount of exposure to feces that our neonates have. And we can vaccinate as well in some circumstances. And the biggest thing we have is removing those shedders. We do our testing and we cull them. And that means we can hopefully reduce the infection pressure on the rest of the herd. But controlling Johne's is definitely a marathon. That's how I explain it to clients. There is not one test and cull all done like pestivirus. This is going to be a long haul situation. And for people who really want to try and control it, they reckon if you do gold standard control for 10 years, you can get a clinically infected herd down to about 3% prevalence, where you won't see any clinical animals, but it will be there grumbling in the background. And if you stop doing your controls, it'll come back. So it's very important that we chunk away and try and make an effort with this disease. So in terms of dairy cows, big thing is testing and finding all of those positive cases and culling them. You don't necessarily have to just cull them and send them away. You can put them into a separate group and fatten them for beef. And if you know that they're pregnant, you can have a little leopard pen. So we can stick them in a pen on their own, away from everybody else, and they can calve and they can poo, and it will only affect them and their calf. It won't be spread to everybody else in the calving paddock. In terms of preventing infection to the neonates, this is one of the easiest things that clients can do. As soon as their calf is born, you take it away from their mother so that they're not going to have a chance to ingest any of that MAP infected feces. And we can give them pasteurized colostrum from negative dams, and we can also give them powdered milk replacer. So we know that there's definitely going to be no MAP in that. Once they're weaned and they're getting towards adolescence, we can send them out onto clean pastures where no adults have been, so there'll be less MAP on the ground. And that includes slurry as well. So we do disperse a lot of our waste manure on ground. It's a great fertilizer, but with that you can take MAP. So you leave it for 6 to 12 months, so lots of UV can get on there and kill the MAP. Hello, Lucy. And then, so hygiene is really, really important. And even for particularly small farms where the same workers will go between all the animals, we're going to try and limit the spread of MAP by cleaning things, having foot dips, maybe changing your overalls between goes from the adult cows to the baby calves, and not sharing around equipment as well. In terms of intrauterine infection, Hannah, we can't really influence that. Unfortunately, we run the risk that we know there's a high chance that a clinical infected cow will have infected her calf already. This is where good records come in use. So if we can write down that its mother was infected, we can monitor the animal, and whether we say we will make you into a beef animal, we won't keep you in our dairy herd, or we will sell you young. It's a decision, but as long as we've got good records, we can then make informed choices about what's going to happen with that animal later on. In terms of preventing infection, we're going to buy from UNIS accredited herds and have property biosecurity. So get a vet in and get them to do a biosecurity audit for you. For beef herds, we can test and cull. Usually for these guys, we'll do an annual blood test, and if anybody comes up as being likely to be positive, we'll then follow that up with a feces test as well to confirm it. Any animals that come up as positive, we can either cull them straight away, or we can put them in a separate paddock, so we call it like a dirty paddock. And those animals, particularly if they're of high genetic merit, can be used for embryo transfer. So we can breed them, flush out the embryos before they become embedded into the uterus and then get the risk of uterine infection. We can then take those out before they're embedded and put them into donor recipient cows. Now that means at least you keep your genetic merit going, and those little calves are hopefully not infected. In terms of pasture management, this is quite important in terms of whether you have all your cows calving at once in one paddock, or whether you can spread them out so they're on a bigger surface area. And so we're reducing the infection load per metre squared. So that's part of your stocking rates, and putting all your weaned calves onto clean, fresh pastures that adults haven't been on for 6 or 12 months. Embryo transfer is really good if you've got particularly small herds that want to get bigger, and you know you've got Yoni disease. You could go headlong into that. But for a lot of our guys, it's quite easy to do Yoni disease accreditation schemes in beef. So we're trying to encourage as many of them as possible to adopt schemes and have a biosecurity plan. For small ruminants, we have a few more options. So we're still going to cull our clinical cases, and we're going to have pasture management again. And if we can rest pastures, that makes life a lot easier. We can have the UV light spreading onto them, killing all those MAP. So at best, we can get 90% of MAP dying each month. So if we leave it at 6 to 12 months, then we've nearly got everything killed. And we can graze cattle on there as well to help mop up some of those MAPs. And in terms of vaccinations, we've got options as well. And for our purchasing, instead of having accreditation schemes, we've got risk-based trading. So you can work out from their vaccination status, their location, the history of units on the farm, and testing as to whether or not they are high risk or low risk. And again, property biosecurity is still important. Jess will talk about our accreditation schemes in 2 seconds. And Sophie wants to know, if you've got a bee farm, you aren't as concerned because they'll go cool before they start showing clinical signs. If you have the adult females and the breeding bulls in the herd, those guys will stick around, and they'll be the ones which will be going clinical and spreading the MAP onto the pastures for your baby calves to ingest. And so yes, a lot of the little ones, they might not live long enough that they'll present clinical signs, but some of them, particularly on the grass-based systems rather than feedlot systems, they'll be living to the point where they might start showing clinical signs. So if you slaughter your steers at 2 years old, they could already be showing clinical signs and losing weight, and so losing your money because you get less carcass value. So in beef systems, it's quite important if you've got you're at the breeder end. And Steph says, if we can't control for the possibility of intrauterine infections, wouldn't putting subclinical calves with non-infected calves negate all these control measures? You're only going to start shedding when they're at least in their teenage years. So we'll find that for a lot of time, it's only the young ones that are really susceptible. They're going to absorb the MAP. So we find that as the young ones get older and are less likely to absorb all that MAP, we then start to find that the clinical ones or the subclinical start to shed. So actually for that cohort of a similar age group, they're not going to cross-infect each other. It's only the old ones spreading it to the young ones that's the problem. So our sheep and goat vaccine is called Gadia. You can't get this everywhere. It's mostly Australian based and New Zealand. It's a single dose of MAP killed in an oil adjuvant. That oil adjuvant is quite important in terms of what signs we see in animals after we've injected them or if we accidentally self-inject. So we inject it behind the ear subcutaneously when we're doing marking, and it causes quite significant lesions at the injection sites. You'll see a big lump and also regional lymph node lesions. If for any reason one of your clients injects themselves with it, they must go to hospital. You can get really, really nasty lesions. The picture down here is actually quite a mild case. There are people who have injected themselves in the leg by accident and sloughed whole heaps of muscle off. It's really not a very nice vaccination if you get it wrong. But it's quite handy in terms of for sheep. If we have animals vaccinated, it takes them, although they might have the disease already, because they ate it off the floor in the feces when they were very small lambs, they will have a slower progression from being infected right through to clinical disease. So they will last longer effectively. It's more of a economic and welfare benefiting vaccine. So it means that these animals live a longer, happier life, a more productive life before they succumb to the disease. So it means we're more likely to cull them for other reasons first. They will still shed the disease, just in small amounts. So it's helpful in terms of control. Hayes put, are vaccine-induced lesions in the animals self-limiting? They tend to get the lesions quite quickly, and then they just kind of become quite quiescent. They just sit there. The abattoirs are quite used to seeing them, and they won't necessarily dock you a lot of money if they've seen these lesions in the carcasses. And they're all around the head, which is why it's really important to get them right behind the ear. Most people are not going to eat the head. So that's why we try and vaccinate them up there, and that's where we're going to get all those lymph node reactions, and we can just bin the head and those lymph nodes. So our industry control schemes, we can have them for all of our ruminants. You won't necessarily find them as part of law in every country, but there are lots of industry-led schemes which people participate in. And it can get you a premium price when you go to sell things. Most developed countries do have a scheme. Unfortunately, the UK and New Zealand don't, because we have Mycobacterium tuberculosis, we have bovine TB, and that interferes really, really badly backwards and forwards between the Johne's test and the TB test. So when we inject them with the reagent to try and induce a response that we can measure, we might find that if we test them too close together, so we do a TB test and then a Johne's test, or the other way around, they cross-react and we get inconclusive results, or we get incorrect results. So we advise that we leave 60 days between doing either a TB test or a Johne's test, or either way around. I used to have a herd back in the UK that I would go and do TB testing on, and several years previously they'd done the Gadir vaccine off-label in the milking cows, and they used to have huge reactions to the TB test because they still had antibody titers to the Gadir vaccine, and it made trying to read their test and interpret it very, very difficult. So for a lot of our clients, education is essential, and trying to give them the best possible plan ahead with the disease. These are all voluntary schemes, and they've got to really get on board with the idea that it's great for welfare and it's great for economics as well. Because testing isn't free, but if it means that you have longer living, healthier livestock, then they'll be able to produce for you. So we said before, our tests are pretty rubbish, but the more we test, the more we can improve our sensitivity. And we can try and overcome the boundaries of a long incubation period of this disease. So we try and test at least annually, and for all of our adults. So we've got check tests and HEC tests as part of our industry streams. So for a check test, we blood sample 50 adults from the herd, or a proportion of that's representative. And we usually start off with our blood ELISA, looking for antibodies. Anybody who is either inconclusive or positive will then go on to having a fecal culture or a high throughput Johne's PCR to confirm the result. Obviously our culture is going to take a lot longer. So during that time, you might wish to isolate any inconclusives or positives from the rest of the herd. For dairies, we're going to do our HEC yard scrape. So HEC is herd environmental culture test. So at milking time, once all the cows have been through the collecting yard, we'll go with a big spade and make a cross across the whole of the yard and take a poo sample all the way from one side to the other, and submit that as a representative sample of everybody in the herd. In terms of official recording, they will only say it's definitely positive if it's confirmed by 2 tests. If you only get 1 positive, then we can't say whether it's right or wrong. We know these tests aren't great in isolation. So here's one of the accreditation sample matrices. So depending on how many animals you have, will tell you how many that you need to test. So this is for a general herd test. This is not for a check test. So if you have a beef herd of 220 animals, then you'll have to test 217. It's very specific. It also gives you an idea of the ages. So for this one, it says 2 years. And subsequent tests, if you know that you haven't got so many cases, you can go to 4 years of age and over. Depending on where you'll work, they have different cut-offs for the ages that you're meant to test. This is the dairy score that we're currently using. All of the accreditation tests in Australia were updated in 2017. So we have new scorecards. So we're going from the very bottom, where you have no idea whether your herd has it or not, right to the top where we know they definitely haven't and they receive regular monitoring and biosecurity. And same for beef. We have unmanaged and unknown risk, all the way up through biosecurity plans, testing, frequent testing, and vet checks, right up to the highest where we know that we are very unlikely to have MAP. In terms of economics, through losses in milk and meat and early culling, we can have lots of monetary loss. So for beef farms and dairy farms, there's quite a lot of money lost per year. This is on an average size farm, and sheep, we have it per hectare. So for these, it's quite a concern for all of our clients. Nobody wants to see animals with poor welfare or that are dying slowly and painfully. And no one wants to worry that their finances are also impacted, because this disease is going to go on for years and years. These figures are not going to disappear overnight. So it's a very big stress for a lot of our clients. And also the danger of self-administration with the vaccine as well is quite a worry. You can imagine if half your legs sloughed off, you're not going to be able to work for a while as well. That's another stressor. One of our concerns for public health is that it has been associated with MAP, with Crohn's disease. Now, it's not been proven, but it is something that we're going to have to keep doing research on and make sure that we have the best information possible so that we can control it as best we can. At the moment, quite a few of the standard pasteurization techniques are not high enough to kill MAP, but they will kill bovine TB. And so we have to think about, is this going to be a human health risk, particularly for things like if you're going to drink raw milk, which happens on farms, and also if you are in the middle of nowhere and you're drinking from creeks and rivers, you're at risk of getting it as well from that contamination side of things. Heidi, there is only one vaccine available. That's it, and we import it around the world under special license for other areas, and they're the only ones that have cornered the market. So in terms of how do we discuss this with our clients and the general public, we can look at, is Crohn's disease and MAP linked specifically, or are they innocent bystanders? They're very similar in that they have both produce a granulomatous infection, and but in terms of the actual pathology, they're quite different. Humans will respond to antimicrobials, but the ones we use are quite broad spectrum. So they can be killing other diseases which are causing comorbidity. MAP can be present in some cases of Crohn's, but control people who don't have Crohn's can also have MAP, and vice versa. So they don't necessarily stand on their own. And yes, MAP DNA can be present in some lesions, and patients can have antibodies to it. But we know our tests are pretty rubbish. So we need to be able to confirm it different, which we haven't yet managed to do. And yes, we might find MAP in milk and water because we're not able to always successfully pasteurize it. But there are places where we know that Yoni disease is absent. So Sweden is a good example of that. And they still have Crohn's disease in local people who have not been to other countries. There's a few references there for you. I'm just going to go through a few questions before I finish. Down we go. Yeah, so unfortunately the company has got a monopoly on the prices. It's not horrendously expensive in comparison to long-term reduced production. So we do find that people do use it. Yes, it's licensed just for sheep, but people have used it in the past off-label for cattle. So back in Victoria, they do use it off-label, and it does have some efficacy. You just have to bear in mind that there are big licensing rules, and in terms of testing, it makes testing much, much more difficult because you've already got those antibodies in there. So really, you can only go down to your fecal tests for PCR and culture. And in terms of MAP in humans, we don't see any clinical disease, asymptomatic. It's our concern that it's linked somehow to Crohn's disease, which is our worry. We can't prove it yet, but one day we might have a better answer if we keep looking. So in terms of when we speak to the press and the public about it, we have to be quite careful that we don't say this is definitely X, Y, or Z, because it's a very difficult disease to test for, and we've still got a lot to learn. Cool, right, has anybody else got any questions? Because I need to let you go to your next set of lectures. But I will try and re-record the pesty one, and my doggy's gone outside, she's gone for a run, she's probably eating wombat poo again, so. Such a nice dog. Thank you very much for all being up this very early morning. I hope you have a lovely Friday and enjoy yourselves, and have a little wave to all your pets as well at home. Okay, see you later.